Welcome to Bio 5A Finals Review. This is Zainab, your supplemental instruction leader. We are trying to do our best to still provide you guys with review material as we transition to online instruction. So I really appreciate everybody coming out and trying to, figure it out as we do as well. So this is for Dr. Dehesh and Dr. Martin's Green's section of Bio 5A. As a lot of you know from the announcement that Dr. Martin's Green made, your final will only include material that Dr. Martin's Green went over. So for this series of review videos, I'm going to be focusing on her material. If you have any questions on Dr. Dehesh's material, or for that fact, even Dr. Martin's Green material, definitely interact in the comments section. I will be there for 2 hours, 12 to 2 this Sunday, to answer any questions and involve and facilitate a discussion. So definitely comment down below, answer each other's questions. Your peers are the best way to learn. Um, also, please sign in. There is a link in the description box to sign into this review session. It's really important that you guys sign in. Please sign in. It helps us keep track of how many of you are attending, and helps us show the importance of these resources so that we can continue providing them to you. So definitely sign in, like the video, comment down below if you have questions, and definitely put in the comment box. But if you don't feel comfortable, then email me and I will put in the comment box, and we can work together to figure this out. So I'm gonna get started with the material. So the first thing that Dr. Martin's Green covered was cell communication, right? And we know that there are several different types, and it's important to kind of understand which of these communication types are an umbrella term and what falls underneath each one, and what distinguishes each type of communication. That being said, the way this can be applied to science, and the way you will probably be tested on this material, is application based, right? So you're thinking about different hormones or different interactions that can take place, and categorizing them in each category accordingly. So if we're talking about hormonal interactions or hormonal signaling, then you would know that it would be long distance, right? And if she gave you, or if you were given a question that was describing this interaction, you would be able to identify it as long distance. So I'm just going to go over kind of the basic definition of each, and if you have any questions, definitely, again, put them in the comment box below. So we're going to talk about local signaling first. So local signaling, as is implied by the name, is signaling with cells that are near each other, right? And autocrine signaling is kind of unique in that it is not a cell that is near the original cell. It is the actual cell that is releasing that ligand that is then um, also having the ligand bind to the receptor. So I said that kind of weird, but what I mean is, in this case, you're signaling to yourself. So you have a cell, it releases its ligand or its signaling molecule out into the extracellular fluid, and then that ligand binds to a receptor on the membrane of the same cell that the ligand was released, as depicted here. So that is signaling to yourself, and autocrine signaling. Next, I'm going to talk about paracrine signaling. So paracrine signaling is signaling to, again, a local cell that is near you. So this involves a secretion of a ligand, a signal molecule again, and a target cell that is really near, in the same proximity, so often in the same tissue that the signaling molecule is, will have a receptor, and that ligand will bind to the receptor and transmit whatever specific signal it can be. And just to emphasize, like, these are very general types of signaling. Um, when we're talking about specific molecules, or if you're looking for a specific reaction, or a specific cellular response, then you aren't really referring to like these general umbrella terms, but those different specific types of cellular response and ligand systems can fall in these overall terms. So then next is very similar to paracrine signaling is synaptic signaling. And synaptic signaling is basically happening in neuron cells, which are the cells in your brain tissue and part of your nervous system. And as you can see, this signal interaction happens at your synapse. So the signaling cell releases its ligand, and then the nerve cell that is adjacent to that original cell will uptake the signaling molecule, and it'll create a membrane potential, which you will learn about later, and that will then, as you can imagine, signal to a neuron that might be right there, right? The next type I'm going to talk about is direct contact. So when you have direct contact, you can have cell-cell recognition or cell junction. So let's start with cell junctions. Cell junction is signaling that is happening without that signal molecule leaving the cytoplasm. So there are tunnels or gap junctions in plasmodesmata that facilitate the movement of signaling molecules between two neighboring cells without it leaving the cytoplasm, right? So it can go through these little tunnels. So in animal cells, those are called gap junctions, and in plant cells, those are called plasmodesmata. And then cell-cell recognition is unique in that we aren't really referring to a signaling molecule or a ligand being released by a cell. I kind of like to think of this as two cells shaking hands. So they're not necessarily passing a note, but they're shaking hands, letting each other know that they're there through their membrane proteins. So those membrane proteins bind together and then probably invoke a cellular response that is specific to that cell and that ligand system, or that, I can't think of the word, that receptor system. Okay, we're going to talk about is long distance signaling, and this is generally specified to endocrine signaling, which is hormonal signaling. And this is when you have a secretion of a signaling molecule from a gland generally, and that signaling molecule will travel through the bloodstream to a different part of the body, where that molecule will then bind to a receptor in a targeting cell. So long distance signaling, you can remember that it would have to travel through the bloodstream, and unlike all of these other types of signaling, it is happening uh, not in the same tissue, not in the same area. It is interacting in a larger scheme transmission pathway, if that makes sense. Um, Okay, and receptor systems, specifically G protein coupled receptor. This is actually a really interesting receptor system, considering that about 25 to 30% of drug development targets this specific signaling system. So it's very pertinent to modern day drug development. So when you're thinking about this analytically, you have to think about where in the transmission pathway you are having drug development attack and disable a whole protein or a whole signaling molecule, and the higher up in the pathway you go, the more you're going to disable, right? So if you're disabling G protein up here at the top of the pathway, then all of these following steps will not happen. So the variety and the different cellular responses that can come from it, out of all of them will be disabled, versus if you're disabling further downstream, like protein kinase A or other signaling molecules down here, then you're attacking a more specific cellular response, so you're better able to tailor to what specific outcome you want from that drug. So specifically, in understanding this system, you need to know that a ligand comes and binds to G protein coupled receptor, and this causes G protein to detach from the coupled receptor and interact with adenyl cyclase, which is another membrane protein. And here, ATP activates CAMP to interact with protein kinase A and invokes cellular response. And the cellular response of this case is glycogen breakdown. So as you guys know, glycogen is the long chain of glucose molecules, and glucose is the base for cellular respiration to produce ATP for all cellular function, right? So this basically is, in general, just about energy production. So this is telling the cell to wake up and to start being active in whatever physiological scenario you are in. So the example Dr. Martins Green likes to give is when you are scared, you have all these endorphins in your body, and those can act as a ligand that binds to the coupled receptor to invoke a flight or fight response, right? So that's really interesting and pertinent. The next type of receptor talked about in this course is the tyrosine kinase receptor. And as you can see, this receptor involves two signaling molecules, and they're the same signaling molecules. And there is an inactive system and an activated system. So with the presence of the signaling molecules, these two membrane proteins form an activated tyrosine kinase receptor, a phosphorylated dimer. A dimer just means that these two parts have now come together and interacted, and ATP is going to provide the phosphates that attach to this receptor and is going to cause this to like kind of cross over, if you can follow my mouse. And this then causes activated relay proteins to come and bind to this membrane receptor, which invokes a cellular response. So in this case, the important thing to identify is that the tyrosine kinase receptor kind of claps its hands together to invoke cellular response, and this is activated by ATP. The next type is fairly straightforward and very general, is your ligand-gated ion channel receptor. So a ligand-gated means, again, that a ligand is the binding of a ligand is going to determine if these ions can come through or not. So in this case, you have a signaling molecule, a ligand, come and bind to this ion channel, and that is kind of like a gatekeeper. Once it's bound to the protein, it'll allow that channel to open. The opening of the channel will allow the ions to rush in, and then when the ions are all inside, the gate is going to close and that signaling molecule is going to leave its binding site. And those signaling ions that went through will invoke a specific cellular response. And then the last type of signal receptor system you need to know is steroid hormone intracellular receptors. So this is really unique because before we were talking about the receptor being a membrane protein, and in this case, your receptor protein is actually inside the cytoplasm, right? So you have a hormone. In this case, it is depicted as testosterone, and testosterone is going to go through your blood, right? So it's excreted by a gland and it travels through your blood, and then it enters the cell, and as you can see, it's just entering the cell. There isn't really any active transport that's taking place. So for that molecule to go through the membrane, it would have to be hydrophobic, right? So that is something that you should know as you're kind of keeping track of where this is going through the cell. So your hormone is going to enter the cell and it's going to bind to the receptor protein and create this hormone receptor complex. And that hormone receptor complex is actually now going to act as a transcription factor. So it'll enter the nucleus through the nuclear pores and bind to DNA to start the production of mRNA and overall protein synthesis. So a new protein is going to be formed. And depending on whatever hormone you have enter the cell and bind to the receptor, you're going to get a different type of protein. That concludes our review on cell signaling systems and receptor ligand systems. Comment down below if you have any questions or concerns, any discrepancies. Like this video and subscribe. The next video I'm going to talk about meiosis and mitosis, so definitely click on over there to join us in that discussion. Don't forget to sign in the link below, and thank you for joining.